All right, so I'm very excited to introduce Todd Macfarlan, who's the head of the section on mammalian epigenome reprogramming at NICHD. So Todd got his PhD from Penn in the lab of Debrabada Chakravarti, and then did his postdoc with Sam Pfaff at Salk. And started his lab in 2012 as a STADMIN investigator at NICHD, and has done basically a series of groundbreaking studies on looking at the relationship between retroelement control, genome evolution, and early development. So he really touches on a number of topics that are of interest to a variety of people in our department, and of interest in general. He's had a flurry of activity in his own lab. So I think there was a 12-month span where you got Nature, Science, and Cell paper, kind of one right after another, and just generally produced a variety of really interesting studies, including the discovery that a family of cripple-associated zinc fingers kind of patrols retroelement expansion in the genome, and there's this really cool sort of evolutionary arms race of the expansion of this family of transcription factors and the expansion of the retroelements. And today I think he's going to actually talk about some germ cell biology, which is very exciting for me. So I'll turn it over to him and let him get started.
Thank you very much, Bluma, for that introduction. I didn't originally intend to work on meiotic recombination, so I'm going to spend just a couple of slides talking about sort of like the lab's primary interest, and then hopefully I'll dovetail a bit into how we got interested in recombination, which really stems from our work on these crab zincfinger proteins. So, as Bluma mentioned, so I'm at the National Institute of Child Health and Human Development, and the central mission of the NICHD is this lofty goal of ensuring that every person is born healthy. But as mammals, we're sort of unique in that we have this unique process of viviparity, or live birth, where the fertilized egg is actually retained within the mother until birth. And obviously this required numerous physiological adaptations to the embryo, including the evolution of organ like the placenta, for example. And the central hypothesis in our laboratory is that this required certain genetic changes. And the idea here is that that we think that retrovirus invasions of the germline in the form of endogenous retroviruses actually played a critical role in both the early as well as the ongoing evolution of placental mammals. And there's some really good lines of evidence that suggest this. The first is that if we actually look at a retrovirus structure, a typical structure, you have these long terminal repeats on the 5' and 3' end, and then you have coding genes for GAG, pol, and envelope, the classic simple retrovirus. There's very strong evidence that proteins within mammals, like the syncytin genes, which are derived from envelopes of retroviruses, are actually essential for placental development. So if you get rid of these syncytin genes, the placenta essentially fails to form, and you have a failure in placental development. So this is just one example describing how retrovirus-derived proteins are actually essential for mammals. But we also know that there's a wide array of regulatory activities from endogenous retroviruses that are present throughout mammals that have been co-opted in numerous cases to drive new expression patterns of various genes. And if you look in the literature, there's a lot of examples of this, but we described a recent example of an LTR that drives some specific genes in human placenta. So you can find lots more of these things, but this is the sort of idea that retroviruses also provide novel regulatory potential. But what I'm most interested in the lab is this other interesting side effect of having these retroviruses infiltrating mammalian genomes, which is we think this has actually led to this expansion of this protein family called the Kruppel-associated box zincfinger proteins, or CRAB ZFPs. And this is a figure we've taken from a recent review we just published in Nature Reviews Genetics, in which you simply just looked at the different genomes of different species throughout vertebrate evolution. And you can count up their different types of repeat elements within their genome. genomes. And the one in red here I'm highlighting are these long terminal repeat or endogenous retroviruses. And you can sort of see here that in marsupials and in eutherian mammals, there's a relatively larger content of these endogenous retrovirus sequences. And this really coincides with the huge expansion of these genes, these so-called crab zinc finger protein genes. So this is basically a chart that counts the number of crab ZFPs in different species. And you can see that there's not that many of these, for example, in birds and in some egg layers, but there seemed to be this big expansion of crab ZFP genes in most mammal species. And what I think is even more interesting, if you look more closely at the number of crab zinc finger genes in each species, what we also see, whoops, sorry about that. What you also see is a relatively tight correlation with the number of zinc finger genes and the number of distinct LTR retroelement families. This was published back in 2011 by Thomas and Schneider. And this really sort of initially led to this idea that perhaps the zinc finger genes are responding, if you will, to retroelement invasions. So what are the crab zinc finger proteins? Well, this is just a slide that highlights the two key molecular features of crab ZFP genes. The first is the crab domain. Many of you might know this as a very potent, strong transcriptional repression domain. And how the crab domain works is it interacts with a protein called CAP1. It's also known as TRIM28. And this actually leads to the recruitment of a series of enzymes, but perhaps most notably, it leads to the recruitment of this enzyme CETDB1, which catalyzes the methylation of lysine 9 on histone H3. And this can essentially establish potent transcriptional silencing. And that's why people have recruited crab domains to Cas9 to induce, stable silencing of your gene of interest. Now, the other key domain of the crab ZFPs that make them quite unique is its modular DNA binding activity. And here I'm just showing you a schematic of a multi-fingered protein, with each finger being an array of amino acids here. And what we've done is we've highlighted these 4 amino acids per finger that we refer to as the fingerprint amino acids. And what these fingerprint amino acids do is control the 3 nucleotide specificity of each zinc finger. So you can essentially build a zinc finger protein that can bind to any sequence that you want. And in fact, before there were CRISPR-Cas9, you might remember that there were zinc finger nucleases that were based on this technology, that you could perhaps design zinc fingers that would interact with specific DNA sequences. So these proteins were really thought to be sequence-specific DNA-binding transcriptional repressors. And this evidence has largely been borne out. So if you knock out crab zinc finger proteins, or if you knock out the co-repressor of crab zinc finger proteins, you generally lead to reactivation of target sequences. So this is just a look at what sort of the lab has been doing over the last 8 or 9 years since I started my group. This was a slide that was generated by Anna Sempt in my lab, taking advantage of data that was published by Michael Imbault and Didier Chrono's lab, in which they actually looked or tried to identify orthologs of crab ZFPs across species. So what this plot is here is it's basically each line here on the vertical axis represents a single crab zinc finger gene that was found in the mouse. And then what we've done is asked, is that specific crab zinc finger gene found in other species across vertebrates? And when you do this kind of analysis, what you find is that there's a very large fraction. So more than half of the crab ZFPs in the mouse genome are completely restricted to the mouse. So these are very young crab ZFP genes that have shown up very recently in rodent evolution. You then have another smaller set that I'm not even highlighting here that are maybe found in a couple of other rodents. And then you have this other core set of mammalian conserved crab zinc finger genes. So you can see these are essentially present in most mammals, but they're not present in egg layers or in chickens and birds. This is a very interesting subset here. And what the lab has essentially been doing is doing functional genomics kinds of studies where we make mouse knockouts for these proteins. And what we've largely found is that when you knock out these crab ZFPs that are present restricted to in just the mouse, is that you get reactivation of retrotransposons, but that these are generally non-essential. The mice can survive without individual crab ZFPs, and in a handful of cases, we've been able to actually knock out entire clusters of crab ZFP genes. So far, we've knocked out about 100 of these, and all the mice so far are generally viable. So these are non-essential transposon suppressors in the mouse lineage. Now, we've also published a couple of papers on some of our favorite mammalian conserved crab zinc-finger proteins, that is amongst this set. And so far, all of these have been essential for development. And just giving you one example of this gene, ZFP568, which is a very potent repressor of a single gene in embryo development called IGF2. So we can essentially think of these crab ZFP genes as being very sequence-specific transcriptional repressors. Okay, now this is just sort of an introduction into where the lab started, but today I'm going to tell you a story about a very unique crab zinc-finger gene. And that is when you look at the ones that are even more conserved than these mammalian conserved ones. And this gene called PRDM9 is the origin of the crab zinc-finger protein family. So, but the PRDM9 gene is quite unique from the majority of crab ZFPs in a couple of distinct ways. The first way that the PRDM9 gene is unique is that its crab domain does not interact with the co-repressor protein CAP1. So if you just do protein interaction assays, it does not interact with the CAP1 co-repressor. The second thing is that PRDM9 has its own PR set domain. This is a histone methyltransferase domain, but it's also quite unique in that it catalyzes the methylation of two distinct lysines on the histone H3 tail. It catalyzes the trimethylation of histone H3 on lysine 4, as well as the lysine 36. And if you knock out PRDM9, you essentially lose both K36 and K4 methylation at target sequences. Now, PRDM9 is similar from other zinc finger proteins in that it also possesses a C2H2 zinc finger array. Now, this is one of the ways in which the PRDM9 is unique from other methyltransferases, and that most methyltransferases don't have direct DNA binding activity. But PRDM9 does. It has this very large C2H2 zinc finger array, which encodes a highly sequence-specific DNA binding protein. Now, it's a bit, what also separates PRDM9, however, from other zinc finger proteins is that it has this highly satellite-like zinc finger array. And then if you actually align each of, let's say, 10 zinc fingers on this one particular PRDM9 allele, you see that each finger is an almost identical replicate to the previous finger, with the exception of a few places within the finger that encode the residues that actually coordinate the DNA binding activities of that finger. So the fact that this protein is such a satellite-like zinc finger array has actually led to extraordinary molecular evolution of the PRDM9 gene. So if you, for example, look within human populations at the zinc finger array of PRDM9, what you can see is that there are many, many alleles within the human population. So each one of these will encode like a unique DNA binding activity for PRDM9. We can also see the same thing in the rodent lineage. At the time this paper was published in 2010, there were something like 60 distinct PRDM9 alleles in the rodent lineage. And we now know that there's many dozen more in the human population. So this makes this a really unique gene. It's a protein coding gene that has a satellite-like sequence. And changes in the satellite sequence actually change the DNA binding activity of PRDM9. Okay. Now, there's one other really key feature that distinguishes PRDM9 from other crab ZFPs, and that is that PRDM9 is expressed almost exclusively in meiosis, and it has this very unique role in defining the sites of DNA double-strand breaks that essentially lead to meiotic recombination hotspots. So the way we think of how PRDM9 functions is it uses this rapidly evolving zinc finger array to bind to DNA. And then via its set domain, it catalyzes the methylation of lysine 4 and lysine 36. And somehow this leads to the downstream generation of DNA double-strand breaks. And this was one of the questions that we were interested in addressing is how does this PRDM9 gene function? Okay, so my Zoom goals today are to provide some background on meiotic recombination, why it's important to study. The second is to describe sort of these extraordinary features of the PRDM9 and how it specifies meiotic hotspots. And then I'm going to spend most of my time discussing our identification of these two proteins called CWPW1 and 2, and how they co-evolved with PRDM9 to change meiotic hotspot patterns in vertebrates. And I should point out, if anybody wants to interrupt me, I'm more than happy to take questions throughout the seminar. So first, just to give you a little bit of introduction on meiotic recombination, why it's an interesting and important phenomenon to study. So meiotic recombination is essential for generating genetic diversity in sexually reproducing organisms. But it's also absolutely essential for the proper alignment and segregation of chromosomes. And I'm going to show you some slides on the next one that talks about the implications in human fertility. Now, mechanistically, meiotic recombination involves a programmed DNA double-strand break, and then its homology-directed repair by one of its by its homologous chromosome. And as I mentioned, that meiotic recombination is not a random process, and that the sites are actually selected, and they typically occur in about 2 to 3 crossover events per chromosome, and these occur in these regions that we refer to as hotspots. And the hotspots are classically defined as regions that are enriched. They typically are somewhere around 1 kb in length, where you have a higher likelihood of having DNA double-strand breaks than the sort of background rate that might occur in the genome. Okay, so what are some of the consequences of failing to undergo proper meiotic recombination? Well, 2 that we can think of are aneuploidy and gross chromosomal rearrangements. And I show this slide because of these really beautiful studies that were done in the past year, in which human egg donation was used to look at the rates of aneuploidies in human eggs. And what you can see here is if you look at the euploidy rate in human eggs over the age, you can see that initially when humans are fertile in their young teens, that there's actually extremely high rates of aneuploidy in young women. And then the rates of aneuploidy drop by the time you get to around 25, or you're sort of in your prime. But then aneuploidy rates begin to pick up again. So as you age, you start to see higher rates of aneuploidy. But sort of on average, you can see that about half of all human eggs fail to properly segregate their chromosomes. And this actually leads to very high rates of miscarriage. Now, a second type of chromosome anomaly that happens in human embryos are gross chromosomal rearrangements, or GCRs. And GCRs are also extremely common in human embryos. So if you look, for example, at eggs, there's about 10% of all human eggs carry gross chromosomal rearrangements. This number goes up to about 20% of pre-implantation embryos. And it results in about 1 in 20,000 live births having gross chromosomal rearrangements. And if you notice, well, what happens is that these very, very high rates in early development usually lead to failure of development. And that's why you're only seeing about 1 in 20,000 live births. Now, one of the other really interesting features about these gross chromosomal rearrangements is if you look at where they occur, they're hugely enriched at meiotic recombination hotspots. So there's something about these sites that are being specified for DNA double-strand breaks in meiosis are actually leading to them to being somehow unstable, either in the eggs themselves or during early development. So we would argue that it's very important to actually study the components of the machinery that establishes the meiotic hotspots in the germ line. Okay, so I mentioned to you that PRDM9 is important for specifying meiotic hotspots, but in fact, over evolution, there seems to be at least 2 different ways to specify hotspots. You have species like plants and in yeast and in birds, hotspots tend to be found at gene promoters. And this just shows you the sort of a generalizable map for where crossovers tend to occur. So they tend to occur at the transcriptional start sites and transcriptional end sites at genes. And this is very unique from PRDM9 system in most mammals and in many vertebrates, and that the PRDM9 is sort of decoupled hotspot determination away from genes. So now you have the hotspots being determined by this unique DNA binding protein PRDM9, which is somehow leading to DNA double-strand breaks occurring from sites away from genes. Now, one really interesting thing that happens is if you, oops, sorry. Slide out of place here. One interesting thing that happens if you knock out PRDM9 in species like mice is that DNA double-strand breaks actually revert back to this ancestral state in which the hotspots are found at promoters. So I'm going to walk you through this slide. So if you look in wild-type mice, for example, and you map meiotic double-strand breaks. This is a ChIP-seq experiment in which they map double-strand breaks by looking at the binding of a protein that essentially recognizes the DNA double-strand break. So you can see in wild-type mice, you have these hotspots that are sort of forming away from these gene promoters, which are labeled here in red. But if you knock out PRDM9, you completely lose the hotspots at these places, and the hotspots instead move to gene promoters. So this really sort of shows that a sort of reversion back to the ancestral state. And one of the things that's quite unique about this is that you'll notice is that you have these H3K4 trimethylation peaks that are happening at the PRDM9 sites that are completely lost in the PRDM9 knockouts. But seemingly these H3K4 methylation that's found with gene promoters somehow sort of leads to the retention of DNA double-strand breaks on promoters. But one thing that also happens in these mutants is that these mice are completely sterile. So despite the fact that they generate breaks still at these sites, they're not efficiently repaired. And this basically leads to seminiferous tubules that actually produce no sperm. So these male mice that lose PRDM9 are completely sterile. Okay, so there are many interesting questions that remain about PRDM9 and its role in specifying meiotic hotspots. And I'm just going to name some of these. We've not addressed most of these. The first was, how does the activity of PRDM9 actually override the ancestral machinery that wants to make breaks at promoters? The second is, why is PRDM9 necessary in mice? I showed you that these mice are sterile when you knock it out. And the reason I say this is that we also know that PRDM9 has been lost in many lineages. And a good example of this is in all canines. So canines, all dogs have completely lost the PRDM9 system. And they now generate DNA double-strand breaks at promoters, just like the PRDM9 knockout mice. So it's clearly not absolutely required for fertility. The third question is PRDM9 playing a role in human infertility? And then sort of our penultimate question is, why did this system evolve at all? And did it provide any selective advantages? So I'm not going to address most of these, but these are things that I love to talk about, and I'd be happy to talk about some of our ideas, at least in the discussion session. But we instead decided to work on sort of a more tractable question, which is, are there factors that might actually read these PRDM9 induced histone marks that could perhaps explain how PRDM9 is leading to the generation of double-strand breaks at its binding site? So here is our hypothetical protein that is recognizing the H3K4 tri and H3K36 trimethylation marks. And this was basically the project that was undertaken by Mohammed Majoub, a postdoc in my laboratory who joined a few years ago. So Mohammed's approach was actually quite simple. His idea was to look for factors that might be tightly co-expressed with PRDM9 during spermatogenesis. And at the time Mohammed started, we were very fortunate in that some of the first single-cell RNA sequencing datasets were coming out, and a couple of beautiful ones were done throughout spermatogenesis. So all the way from spermatogonial stem cells all the way to mature sperm. And then he essentially simply designed a computational pipeline that would allow him to look for factors that were tightly co-expressed. And basically, he's just plotting these in a ranked list. Here's the PRDM9 gene, its expression in single cells throughout spermatogenesis. And then he rank sorts all the genes for their co-expression with PRDM9. And the number one hit was this factor, ZCWPW1. And if we looked at ZCWPW1's expression in different tissues, we saw that it is highly abundant in the testes and is expressed at much lower levels in virtually all somatic tissues. And so we generated an antibody against the ZCWPW1 factor and found, again, that it's pretty uniquely expressed in testes. Now, I wanted to make a point here that we were also here looking at ovaries, and these are mature ovaries. But mature ovaries aren't undergoing early stages of meiosis. So we also know that ZCW1 is expressed in the fetal ovary at the time cells are undergoing meiosis. So this seems to be a factor that's pretty restricted to meiosis. So its expression pattern fit with a factor that might be involved in recombination. But we really knew we were onto something when we looked at the sort of domain architecture of the ZCW1 factor. And I'm just going to highlight the two key domains. The ZFCW domain had been shown on a number of proteins to be an H3K4 trimethylation reader protein. And the PWWP domain is found on a number of proteins that possess H3K36 trimethylation reading activity. And this is also, for example, found in DNA methyltransferases. So we, right off the bat, we're thinking, aha, this is the factor. These happen to be the two marks that are placed by PRDM9. We also found another domain on the protein that showed distant homology to a protein called synaptonemal complex protein 1. And the synaptonemal protein complex is a key rigid structure that essentially aligns the chromosome axis on homologous chromosomes during the act of crossing over. So we could sort of build this model that perhaps the ZCWPW1 factor was recognizing the histone marks and somehow binding to the synaptonemal protein complex to allow recruitment to the enzyme that actually makes the DNA double-strand break. And that enzyme is quite conserved. It's called SPO11. So we sort of had our hypothetical link to a factor that might link the PRDM9-induced histone marks to the sites of recombination. So that was the model that we started with. And we decided to test the model doing a number of biochemical and in vivo kinds of assays. So the first question that we asked is the ZCWPW1 factor really recognizing these dual histone methylation marks? So we generated histone peptides that contained methylated sequences on either K4, K36, or both K4 and K36. And then we mixed these with recombinant ZCWPW1. And what you can see here is that ZCWPW1 does not bind very well to unmethylated histone peptides. It binds pretty well to either K4 trimethyl alone or K36 trimethyl alone, but it binds with the highest affinity to histones that have both of those sites on the same histone tail. And then we decided to measure this more quantitatively. So we took advantage of a collaborator, Xiaodong Cheng, who had developed this isothermal titration calorimetry method for measuring histone binding proteins. And what we essentially found was the same thing that we found in this sort of less quantitative assay, which was that the binding efficiency was about at least 10-fold greater for histone peptides that had both the K4 and the K36 trimethylation marks. So even though this factor can recognize both, it has a much higher affinity for the dually methylated histone peptides. So our next question was, well, does this factor actually bind to these dually methylated sites in the genome? So we decided to take advantage of the cut and run method for mapping protein binding to DNA. And we generated a polyclonal antibody against the ZCWPW1 factor. And then we looked in testes to see whether or not ZCWPW1 was actually interacting with meiotic hotspots. So here I'm just showing you 158 KB window on chromosome 4, in which we've, using other ChIP-seq data sets that were previously published, to be able to establish different types of chromatin regions. So here I'm just showing you DMC1, which measures sites of DNA double-strand break, so we can distinguish, for example, a meiotic hotspot or 2 hotspots here shown in red. Now, when we looked for the histone methylation marks, in this case, H3K36 and H3K4 trimethylation, what you can see is that pretty much only at meiotic hotspots do you see the co-appearance of K36 and K4. You also see H3K4 trimethylation at the promoters of genes, and you see H3K36 trimethylation within the gene bodies, but you generally only see both marks at meiotic hotspots in the testes. So when we looked at where ZCWPW1 binds, we see that ZCWPW1 is pretty much restricted to these meiotic hotspots. We don't see it very strongly at promoters or within gene bodies. And if we look at this sort of in a genome-wide pattern for the places where ZCWPW1 binds, about 85% of its peak regions have the co-appearance of K4 and K36 methylation. So it seems to be strongly associating genome-wide with these sites. And from previous data sets, we also know that these are the PRDM9 dependent sites within the genome. And just if we look at this more carefully, we can actually look at the nucleosomal binding pattern of ZCWPW1 within these data sets, because we use this cut and run method, which essentially cleaves between nucleosomes because there's an MNA step. And what you can see here is that when we center the peak regions on the PRDM9 motifs, you see this very beautiful pattern where you have a ZCWPW1 binding to the sort of +1 and +2 and -1 and -2 nucleosomes to where the PRDM9 sort of motif is centered. And this matches quite nicely with the histone methylation sites sort of genome-wide. Okay, so this data really got us excited that yes, ZCWPW1 seems to be very restricted to the sites in the genome where PRDM9 is binding. But we really wanted to determine whether or not its binding was PRDM9 dependent. So ideally, we would have done this experiment again in a PRDM9 knockout, but we didn't have the PRDM9 knockout in the laboratory. So we decided instead to take advantage of another trick. And that is that I mentioned earlier that PRDM9 has a lot of alleles within different rodent populations. So if we take a PRDM9 black 6 mouse that has this specific PRDM9 allele, we could also map ZCW binding in a hybrid mouse or a castaneous mouse that has a completely unique DNA binding activity. So this is what we did. So we mapped ZCW binding to the genome in either a pure black 6 background or in an F1 hybrid between a black 6 and a castaneous mouse. And what you can see here is that ZCWPW1 is still binding to these black 6 sites, but we also get the appearance of these novel sites within the genome, or ZCW binds. And if we look at these motifs, we see that this can totally be explained by the motifs that are bound by those 2 PRDM9 alleles. So for example, in the black 6 mice, we get this single motif, which is the known PRDM9 motif found in black 6 mice. And then these novel motifs that are showing up in our hybrids can basically be explained by the DNA binding activity of the castaneous PRDM9 allele. And just to show you that this also is true genome-wide, you can see that in black 6 mice, we only have ZCWPW1 binding to black 6 hotspots. But ZCWPW1 binds both to black 6 as well as castaneous hotspots in our F1 hybrid. So this really shows you and proves that ZCWPW1's binding to the genome is PRDM9 dependent. Okay, so the next key question is, well, is this factor ZCWPW1 actually essential for meiotic recombination? So we generated a ZCWPW1 null mutation that had an exon 5 deletion. And we can show you here from the genotyping of our wild type and ZCWPW1 knockouts. This was actually just done with Western blotting in this particular case, is that ZCWPW1 is really only expressed in the testes of wild type, but we completely lose expression in our ZCWPW1 knockout testes. Now, these mice are completely fertile. I'm sorry, they're completely viable, and they're born at Mendelian ratios, but that the males are completely sterile. So here I'm just showing you some basic phenotyping of ZCWPW1 knockout males and females. So if you look at the males, they have very small testes, and here you can see a wild type testes in an Eppendorf tube. And here you can see a knockout testes that's much smaller. It's about 40% or so the size. And if you actually look at the adult testes, you see that they're completely lacking sperm in the seminiferous tubules. So they don't produce any sperm at all. And if we look carefully, I'm going to show you some slides. They basically are undergoing meiotic arrest. This is in contrast to the phenotype in females. So in females, we basically see that the ovaries look pretty normal in adults. But if you follow the litter sizes of these mutant females, what we found is that these females actually do have a somewhat reduced fertility. So they produce smaller litter sizes, and they have less cumulative pups over time. And they often miss litters, so relative to their wild types. So we wanted to look more carefully at the type of arrest that occurs in these ZCWPW1 mutants. So one of the first things that we decided to look for was whether or not homologous chromosomes properly synapse. Now, I mentioned to you that one of the key things that meiotic recombination does is it's involved in ensuring the proper alignment and segregation of homologous chromosomes. So to look at chromosome synapsis, there's a very simple co-staining that you can do. You can basically stain meiotic spreads of chromosomes with this antibody that recognizes the SYCP3 protein, which essentially labels all the chromosomes. And you can also label with SYCP1, which forms a bridge only between homolog pairs. And then if you look at the overlap between SYCP3 and SYCP1, you should have 19 chromosomes perfectly pairing with one another. And this would indicate perfect synapsis. The only one that's missing here is the sex chromosome. So the X and Y chromosomes don't pair. But if you look in the ZCWPW1 knockouts, what you find is that some of the chromosomes pair properly, but it's almost always less than the full set of 19 chromosomes. So in this particular case, actually only 15 of the 19 homologous chromosomes successfully paired at this stage in meiosis. So we could then quantify this. So if we look at wild type males relative to knockout males, you see that there's always some synapsis, but it's never the complete set of 19 chromosomes. And we think this is why this leads to sterility in the males. Now, I mentioned to you that the females are not sterile, but we still see a pretty strong reduction in synapsis in knockout females. But you'll notice that some of the females can completely pair their 19 chromosomes. And we think that's why that these are actually still fertile. So the females seem to be able to cope with the fact that there's this sort of sub-pairing efficiency, but they're able to still get, enough of them are still able to complete meiosis and complete oogenesis. So this actually led us to the key question. So this phenotype for ZCWPW1 was slightly less, slightly less of a phenotype than what you see in PRDM9 knockouts. In PRDM9 knockouts, you have sterility in both males and females. So we were essentially asking this question: is the ZCWPW1 factor, is it essential for the generation of DNA double-strand breaks at hotspots? So in our first model, we predicted that in the absence of ZCWPW1, you wouldn't be able to recruit these hotspots to the synaptonemal complex to allow for DNA double-strand break sites. So if this is the case, if we knock out ZCWPW1, we reason that hotspots should switch back to this promoter state. This is what happens in the PRDM9 knockouts. If you don't place these histone methylation sites, you can't make DNA double-strand breaks here. So this was one prediction. And the second prediction, if ZCWPW1 is not essential for the generation of breaks, then you should still see DNA double-strand breaks at PRDM9 sites. So this was the experiment. The key experiment was to ask whether or not ZCW was essential for generating breaks at hotspots. So how we decided to do this was to take advantage of a new method, and I'm just going to highlight this. So how do we map DNA double-strand breaks? Well, the method that I talked about earlier, there's a relatively simple method that was called DMC1 SSDS. This is essentially a chip sequencing experiment in which you map the binding of this factor DMC1, which binds to the DNA double-strand break sites. So this is sort of like an indirect method of mapping DNA double-strand breaks. There had been at least one other method that was published by Scott Keeney's group in which they directly sequence the released oligos that are released after DNA double-strand break sites. But it turns out that this method actually requires a lot of testes material. So we decided to collaborate with Andre Nussensweig lab at the National Cancer Institute, who developed this method called end sequencing, which is really a direct method of mapping DNA double-strand breaks. And in principle, this works by essentially just ligating a linker molecule into these very carefully prepared DNA samples from meiotic spreads, and which you can then map directly the DNA double-strand break sites. And this is the method that we used for our study. So I'm going to walk you through this experiment in a step. So here's what happens in the various mutants when we look at the DNA double-strand breaks. So in wild type's case, we have, if we look at H3K4 trimethylation here, we have a PRDM9 binding site here labeled in orange, and we have a promoter site here labeled in blue. And if you look in the DNA double-strand breaks by mapping DMC1, you see that DNA double-strand breaks in wild type mice are only found at the PRDM9 sites. But if you knock out PRDM9, the DNA double-strand breaks move to the promoter regions. Okay, I've described that a couple of times. This is just one example. So what happens in the ZCWPW1 mutants was a bit unexpected. So we were expecting that in these mutants that the DNA double-strand breaks were going to move to promoters. But in fact, that's not what we observed. What we see is that the DNA double-strand breaks are still happening at these PRDM9 sites, but they get even hotter, right? You actually see the accumulation of breaks. And we think what this means is that the ZCWPW1 factor is involved in their efficient repair. You still get DNA double-strand breaks at these sites, but you fail to repair them properly. So you get these accumulated breaks. So this has sort of led us to look, actually, let me just show you this genome-wide first. So just to prove that that's not just happening at this one particular site. So we see an almost perfect overlap between the genome-wide DNA double-strand breaks between wild type and knockouts. And you can see not even a hint of switching of the DNA double-strand breaks to the promoter sequences, which we observe that in PRDM9 knockouts. So what we believe here is that perhaps what the ZCWPW1 factor is doing is it's binding to the sites where PRDM9 is placing the marks. But it's not important for the breaks per se, but perhaps it's somehow involved in facilitating engagement with the homolog to allow for their efficient repair. And this is really just a model. We haven't proven, we don't have any evidence of this yet. But this really had us puzzled. So if ZCWPW1 is not the factor required for generating double-strand breaks at these hotspots, why is PRDM9's set domain necessary for this activity? Now, I haven't described this experiment, but there was some elegant experiments done by multiple groups that showed if you mutate just the set domain of PRDM9, you fail to induce DNA double-strand breaks at these sites. So you clearly need those histone methyl marks at these nucleosides. So this was puzzling then. So why don't you need the reader? And we thought about this for a long time, and we started to go back to square one again and say, well, maybe we're missing, maybe there's an additional reader. And to make a long story short, I'm going to go through this in a couple of slides, but we essentially found that there's at least one other ZCW-like gene, and this factor is called ZCWPW2. We initially missed this in our analysis of genes expressed in single cells because the ZCWPW2 gene was actually annotated as a pseudogene in the latest build of the mouse genome. But when we looked more carefully, it seems to be misannotated. And in fact, we think that the ZCW2 gene is an intact gene, and I'm going to show you our results that supports this. Now, the key way that the ZCW2 factor is similar to ZCW1 is it possesses the same two histone reader activities, but it lacks this N-terminal SCP interacting domain. So we're not sure exactly how this protein functions, but we presume that perhaps this was the reader that was actually linking the PRDM9 sites to the DNA double-strand break machinery. So first off, I just wanted to show you that similar to ZCWPW1, ZCWPW2, if you look at its expression in different tissues, it seems to also be quite restricted to the testes. And if you look in single cell maps during spermatogenesis, you can kind of see these purple cells here. These are basically, this is the PRDM9 expression is very restricted to the leptotene stage of meiotic prophase. You can see that both ZCW1 and ZCW2 are really largely overlapping with PRDM9. So they're very restricted to the same window of meiosis. So we tested whether or not ZCW2 also binds to histone tails that harbor the PRDM9-induced histone marks. So what we found is that ZCWPW1 and ZCWPW2 both have the same preference for binding to histone methylated peptides that have both the K4 and the K36 trimethylation marks. Now, we're still in the process of generating ZCW2 antibodies. So we haven't been able to prove yet that this is actually sitting on meiotic hotspots in vivo. But we did generate a ZCW2 knockout. And what we found is that the ZCW2 knockout looks even more similar to the PRDM9 knockout, and that you have complete sterility of the males, but you also have sterility in females. So you'll see that both the testes as well as the ovaries are much smaller in ZCW2 knockouts. You completely lack sperm in the knockout adult testes, but you also lack mature follicles in females. So this gene seems to be essential in both males and in females for fertility. If we look carefully at where the arrest is occurring, we see a very similar meiotic arrest in the ZCW2 knockouts, in which you have essentially a failure to undergo complete synapsis. And again, I'm just showing you the SYCP3 and SYCP1 staining. And in this particular example, you essentially see that not all the chromosomes are properly synapsing. The other thing that we looked at is whether or not there are actually DNA double-strand breaks in these mutants. And what you can see by just staining with DMC1 is that the ZCW2 gene is not essential for making breaks because you still have a lot of DNA double-strand breaks in these mutants. So it's clearly not essential for breaks per se, but the key question was, well, where are these breaks occurring? Are these breaks occurring at hotspots or are they happening at promoter regions? So we essentially did the same experiment that I showed you previously for the ZCW1 mutants, but now we did these in the ZCW2 mutants. So I'm going to show you a couple of different experiments here. So the first, again, is when you look at meiotic hotspots. I'm going to walk you through this from top to bottom. So we have two distinct sites that have PRDM9 motifs. So in this one here, you have hotspots that are found in wild types that are completely lost in PRDM9 knockouts. So here, for example, again, I'm showing you that the hotspots are switching to promoter sequences. In ZCWPW1 knockouts, almost nothing happens. The sites do not move to the promoters. But if we look at the ZCW2 knockouts, what you see is a somewhat intermediate phenotype. You lose some hotspots that shift to the promoter sequences, but you still see some DNA double-strand breaks happening at hotspots. Now, I'm going to show you this in the next slide, sort of in a genome-wide pattern. So here, if we're looking at two classes of sites, we have the PRDM9 sites and transcriptional start sites. So in wild type, you have almost all hotspots happening at PRDM9 sites. If you knock out PRDM9 over here, you have a complete switch. Now you have all the hotspots happening at promoters. In the ZCW2 knockouts, you see this intermediate phenotype. Some hotspots are retained at PRDM9 sites, and some of them are moving to transcriptional start sites. So how do we explain this data? Well, I'm going to just share with you our model for what we think is happening. And I think this actually explains a lot of things that have happened over evolution. We believe that there are essentially multiple reading systems that are helping to recruit machinery to the DNA double-strand break machinery. So we think there are likely this ancestral machinery. We have an idea of what this protein might be. This protein is called CXXC1. There's an ortholog of this in yeast, which is essential for making double-strand breaks like promoters. And then we also have this newer machinery, these ZCW factors, and in particular, the ZCWPW2. And this factor is the dual histone methylation reader. So in wild type cells, you have this real preponderance of hotspots that are happening at PRDM9 sites. And that's because ZCWPW2 is really engineered to recognize these sites and recruit that to the DNA double-strand break machinery. Now, if you remove PRDM9, you no longer have any histone methylation happening at PRDM9 sites. So we believe in this setup that the ancestral machinery now becomes active. And it's sufficient to bind to these regions and help recruit the SPO11 enzyme to cut these sites. What happens in the ZCW2 knockouts is that the PRDM9 activity is still present. So you still have the accumulation of histone methylation of lysine 4 at these PRDM9 hotspots. So here what happens is the ancestral machinery can read both promoters and PRDM9 sites. And you get about a 50-50 split amongst these hotspots. Some are happening at promoters and some are actually happening at the PRDM9 binding sites. So just in the last couple of slides, I want to talk a little bit more about the evolution of the PRDM9 system. So when I say it's a system is because we believe that this system only works if you have these three components, the PRDM9 gene, as well as the two reader proteins. So I mentioned to you that the PRDM9 gene first originated in jawless fish. And you can see PRDM9 in jawless fish, other fishes. It's lost in amphibians. It's also lost in birds and crocodiles. If you look at the ZCWPW1 factors, we see that they also are lost in birds and crocodiles. So you seem to have a sort of the loss of, the PRDM9 and the ZCW factors. And if you look, for example, in amphibians, you see that there's a loss of PRDM9, and there seems to be loss in some species of the ZCW factors. Again, suggesting that these things, if they really need to work in meiotic recombination, you need to have all of them present. But you'll notice one interesting exception to this is in bony fishes. So bony fish, some of them seem to have a PRDM9, but there seem to be some loss of the ZCW factors. So we wanted to look at this a little bit more carefully. So we zoom in on a number of bony fishes. And what we found is that there were some bony fish that have both PRDM9 and the ZCW factors. There are some bony fish which have PRDM9, but not the ZCW1 gene. And then there are a handful that don't have PRDM9 but seem to retain ZCWPW1. So we looked carefully at the second subset. These are the ones that have PRDM9 but don't have ZCWPW1. Because we sort of reasoned that these types of PRDM9 would not function without the proper reader proteins. So we looked carefully at the sequence. We looked carefully at the sequence of these PRDM9 alleles. Specifically, we wanted to ask whether or not perhaps these were non-functioning PRDM9 alleles. And we looked specifically at the set domain of PRDM9. There had been some previous experiments that had found that there are some specific tyrosine residues of the set domain that were mutated in some species. And it turns out that if you inherit a set domain that lacks one of these tyrosines or has one of these tyrosine mutations, one of three found within the set domain, it completely kills the catalytic activity of PRDM9. And what we found is that when you look at these group 2 fishes, these ones that have a PRDM9 but no ZCWs, these are all accumulating these catalytically dead forms of PRDM9. So what we think that this means is that if you lose PRDM9 completely, or if you get a mutation of the PRDM9 enzyme activity, you really no longer need these histone methyl reader proteins for PRDM9, which really suggests that these proteins really sort of come as a package. In order for this system to work in meiotic recombination, you need both the histone methyl writer as well as the histone methyl reader. Okay, this is my last slide. So there's still a lot of interesting questions about PRDM9 and ZCW and their role in specifying hotspots. We still don't know how the ZCW2 factor actually facilitates the formation of DNA double-strand breaks and how ZCW1 might facilitate their repair. We're not 100% sure why these are absolutely necessary in mice, because as I showed you, the genes are lost in many species, and they're still able to undergo formation of germ cells. Is PRDM9 playing a role in human infertility? I'm only going to just share this very quickly. We believe that it is, and that's because we've actually looked at a small population of 50 males who had unexplained infertility. And we sequenced the zincfinger array of PRDM9 in these patients. And we actually found 2 totally undescribed novel alleles in these azoospermic males. So we actually believe that perhaps these are dominant negative mutations in men. I didn't get to talk about this at all, but PRDM9 has this interesting role in cancer genome evolution. PRDM9 actually gets expressed in about 10% of cancers across a wide range of tissues. And in these cancers, the genome actually becomes unstable. So you essentially get chromosomal abnormalities at meiotic hotspots in the tumors where PRDM9 gets activated. And I think that's a very interesting sort of phenomenon, but we don't quite know how that occurs. And I guess the last question again I mentioned is, why does the PRDM9 system evolve at all? And we're sort of really interested in thinking about why this system evolved and what type of selective advantage it provided. Okay, I'd just like to thank those that contributed to this work. So the work was really spearheaded and led by Mohammed Bajoub, the postdoc in my laboratory, but he had help from a number of people. Sherry Rawls, my lab manager and technician. We had 3 reproductive endocrinology fellows, Blake Evans, Ann Martini, and Carrie Walker, as well as another postdoc, Melania Bruno, who contributed to this work. We couldn't have done this work without a very nice collaboration with the National Cancer Institute and Andre Nussensweig lab, specifically Jacob Payano, who was a graduate student in his group. And we also got a lot of advice from Dan Camerini at Taro's lab, and specifically I'd like to acknowledge Flo Prato and Kevin Brick, who helped us with a lot of the meiotic work. At this time, I'm happy to stop and take any questions you might have.
All right, thank you. And yeah, anyone who has questions, virtually raise your hands, or you could put it in the chat. I can start out because I have a couple of questions. Sure. So on a sort of more specific level, it looks like the I'm going to butcher the acronym, the ZCWPW2 knockout had a more severe spermatogenesis phenotype than the 1 knockout. It looked like there were some tubules that were completely missing germ cells. Is that fair to say?
So actually, we think that there's complete meiotic arrest and apoptosis in both cases. So it might have just been a specific of that particular subset. There is a clear difference in the female phenotypes, in which, and this is why we think ZCW2 is upstream of ZCWPW1. And we're doing the classic epistasis now, so we have to do the double knockouts. Because the other possibility is that there's some compensation of we actually think ZCW2 works first, because it's the one that has to bind and lead to the DNA double strand breaks. And then we think the ZCW1 is somehow either involved in homology search or synapse formation, and mechanistically, we don't know how that works. We've kind of ruled out a simple model that it dimerizes because it doesn't look like it interacts with itself. But there's this interesting, that if you follow this field, what's become quite clear is that even though we know that the PRDM9 system is leading to a break on one homolog, we know that you only get recombination typically if you also have a PRDM9 binding motif on the other homolog. So you seem to need reciprocal binding of PRDM9 to both homologs. And that sort of makes sense. And we're guessing that you need to have ZCW1 sitting on both of those. And that somehow helps lead to more efficient homology research or some aspect of the repair process. And our guess is that male mice are very susceptible, that if you don't do this quickly enough, it leads to the apoptosis pathways. But that females have a longer period of time in which they can repair, even if they're not reciprocally bound. And that's how we think that the ZCW knockout females can actually enough of those can actually get through meiosis. And our prediction, though, is that probably you'll have altered hotspot usage in those females, which we haven't been able to do yet because it's a difficult experiment that requires us to get our mutant on 2 different genetic backgrounds. So we haven't done that experiment yet. Yeah. Mm.
I'm going to squeeze in one more question, and then I see
Sure.
June has a question in the chat, and Steve
Okay.
has raised hand. But I noticed in your Western furs, you see WPW1 that there was a bit of expression in the thymus, which is interesting for recombination reasons.
Yeah.
Tissue where you might expect something. So I was wondering if there's anything interesting there.
Yeah, so we noticed that too, and you can see it even in the single cell data. And in fact, even PRDM9 has a little bit of expression in some thymic, cells. So you hit on what we think is a super interesting question, because the other thing I should point out is that the vertebrate immune system, so VDJ recombination, also shows up around the time where the PRDM9 system comes up. But it actually the RAG recombinase, this gene that's involved in VDJ recombination, actually comes up a little bit after immune regulations. What I mean by this is that there are hagfishes as a species of fish that has recombination but doesn't have RAG. It doesn't use the RAG recombination, and it doesn't use immunoglobulins. It has a different type of recombination system. They're called the TLR system. It's these, it's the VLRs. They're called variable lymphocyte receptors. So it's a totally different type of recombination that's not RAG mediated. So we started reaching out to the people who work on hagfish because we were wondering whether the PRDM9 system might be the system that is the ancestral type of recombination even in the immune system. And then perhaps that residual expression is still around. I mean, so we haven't looked at like immune, repertoire yet in our mutants, but certainly that's something that we're interested in doing, is to see if it might have a little bit of this role in efficient recombination, let's say, but we haven't done that yet.
That's very cool. So June has a question in the chat, and June, if you want to read it, you can jump in. Otherwise, I'll go ahead and read it.
Yeah. It's a great talk, Todd. I really enjoy it. So I just have a question about the binding specificity of the ZC proteins, right? ZCW is something I had a hard time to figure out, remembering it. So if you think about the H3K4 methylation and the H3K36, you can still, although one is concentrated on promoters, one is on team body, you can probably still see some overlapping to some degree potentially. And I'm thinking, whether it's possible that there's a certain level of specificity that's conferred by recognizing both markers on the same histone tail versus on the two histone molecules within the same nucleosome core or between the different neighboring nucleosomes. I just wonder if you have a chance to look into this.
Yeah, so this is to me, this is like the question for the biochemistry people. So we've started to me, what we need to do is mass spec those histones that it's bound to, right? To see if it's on the same tail. I think that really hits at the same question. And we don't know yet, but we suspect that hotspots might be unique in being the only places where it's on the same tail. So that's hypothesis one. Hypothesis two is that there's additional machinery helping to recruit it to the meiotic hotspots. And the one question we get a lot is, well, does PRDM9 maybe directly bind to the ZCW factors? We detect no hint of interaction between those proteins. So, we sort of favor the idea that you can separate PRDM9 and ZCW physically by just having a really efficient interaction with the marks. But we haven't proven this yet. And, it's difficult. That these mass spec experiments are difficult because you would need to isolate a very small number of histones from a cell. And then, you have to, typically people use trypsin to digest, but to get a K4 and a K36 on a same mass spec read is difficult because you typically are chopping up in between there. So, we're trying to develop a method that addresses this exact question, but we don't have an answer yet. Yeah. But what I can say is that, like, if you look, there are some immune cells that express ZCW1, and we can detect ZCW1 binding to the genome in the cells that essentially don't express any PRDM9. So we think there's probably some background binding in those kinds of cells, and we do see it at promoters in that case. You also see, if you massively overexpress ZCW1, that it can bind to promoters. So, our guess is that it's all about, keeping its levels low enough in spermatics, in meiotic cells, that at the levels that it's expressed, it really prefers the dually methylated sites. That's our model. Yeah.
I think Steve Riley had his hand up for a while. I don't know if it's
great talk. Maybe I misremembered one of your slides, but I think I saw there being like maybe 1,000 or so potential hotspots that displayed the these dual methylations, but then there's only 2 actual recombination events per chromatid. So do you have a sense of what's causing that choice? Is it the strongest ones, the ones that are rightly spaced? And then does that change in your mutants?
Yeah, so that's a good question too. So really, the more hotspots, the more deeply you map hotspots with any method, the more hotspots you find. You know what I mean? So in other words, and in the experiments that we did where we're taking a single testes, we're only catching the hottest hotspots. So it's quite clear that if you look at what controls how hot something is, it's basically all about PRDM9 binding, PRDM9 induced methylation. So like the most highly methylated sites, the most highly ZCW recruited sites are also the ones that get the most DNA double strand breaks. Now we also know that, when I say we, the field knows that you always make more breaks than you have recombination events. So what happens is there's a lot of mechanisms. So you might make 10 breaks on a chromosome, only 2 become recombinants, but the other one get repaired by these mini gene conversion events. So not all of them become complete recombination events, but they have little hallmarks of recombination. So you always generate more breaks than you actually lead to full recombination events. And there's, my lab does not work on this. There's a lot of labs who work on, this is all like classic meiotic recombination field. Like we've really stuck to like just the upstream part about like what's leading to the breaks at these sort of sites. But yeah, I think your question is spot on. And there are people who is really working on that. Yeah. Mm-hmm.
And Nina has a question in the chat. Nina, do you want to read it?
Sure. Let me see if I can find the chat here. Hold
on. Can you hear me?
Yes, I can hear you. Great.
Okay, great. Yeah. So yeah, my question was, you mentioned that PRDM9 seems to be expressed in many cancers, and that those cancers often have genome instability. Do you have a sense whether it's that expression that drives the genome instability, or maybe, or is it a consequence of general weird gene expression?
No, so I would say that we're pretty certain that PRDM9 is driving that instability. And the reason is, if you look at those cancers where PRDM9 is expressed, and you map where the instability happens, it's hugely enriched at PRDM9 motifs, right? So in other words, the instability is happening at the places where the PRDM9 is binding. Now, one thing that it's not clear is that it's probably not a direct recruitment to SPO11, which is the enzyme in meiosis that makes breaks. The reason is that those cancers do not reactivate SPO11. So what I think is happening is that just the PRDM9 enzyme activity, so just by methylating those histones with K4 and K36, is probably just like opening chromatin a bit. And somehow, with all the other processes that are going wrong in cancer, it leads to them being slightly more unstable in those regions. This is something we're actually hoping to test, but it's very simple. You could just overexpress PRDM9 in culture, with and without its enzyme activity, and prove that it's due to enzyme activity, for example. But no one has done that. It's a relatively simple experiment, but no one's done that yet. Yeah. Mm-hmm.
Great, thank you.
Mm-hmm.
Any other questions? Okay, I guess if there are no other questions, then thank you again for a great seminar, and thanks for coming. And
you're welcome.
Have a good rest of the day.
Thank you. I'll see you. Bye.